Okay, in this lecture series, we're going to be discussing nutritional physiology of fishes and metabolic processes, and we're also going to spend a little bit of time talking about bioenergetics. Before we get into our discussion of nutritional physiology in fish, I think it's important to recognize that fish are perhaps the most diverse class of monogastrics. Monogastric means one stomach, and this is a term that differentiates fish and other monogastrics from polygastric species, species that have multiple stomachs. They're also referred to as ruminants. Cows are a really good example of a ruminant species. They have multiple compartments within their stomach, and the intestinal system of these species functions largely to house microbiota that do much of the digestive work for them. So fish are not ruminants, they are monogastrics, so they don't have a rumen, although they do have a substantial number of microorganisms that colonize their intestinal system as well to help facilitate various digestive processes. But again, fish are monogastrics, and I would argue that they're the most diverse grouping of monogastrics. If there is a food resource out there that has some nutritional value, odds are there's a fish species that specializes in consuming just that. So we have omnivores that eat all sorts of different things. We have carnivores, herbivores, planktivores, invertivores, detritivores, species that specialize in consuming strictly the scales of other species, species that focus on consuming blood and bodily fluids of other species. There are lots and lots of different types of feeding strategies in fish, and with this diversity comes a substantial diversity in the morphology and function of the gastrointestinal system, which we will discuss in just a moment. Of course, part of a feeding strategy is food or feed acquisition. Now, generally speaking, we think of food. When we use the term food, we're talking about human food. When we talk about feed, we're talking about animal food. So you'll see food and feed used somewhat interchangeably in this lecture, but understand that when I refer to feed, I'm not talking about exclusively pelleted animal feeds like we use in the aquaculture industry. So with respect to feed acquisition, the first part of that is detection mechanisms. How do we know where that food item is? How do we know how to acquire it? So there are a couple of different strategies. Visual detection mechanisms are, of course, very, very common in fishes. If you have an organism that is primarily acquiring its food through visual detection mechanisms, they will tend to have large eyes, like the fish that you see here, but they will be frontally oriented. So you can see this great white shark, you're looking at the front of the fish, and you can see both of its eyes. That would not be the case with other species that have laterally oriented eyes. Fish with laterally oriented eyes, they may be visual predators, but more often than not, they are the prey. And so their eyes are located on the sides of their body to increase their field of vision and hopefully avoid predation. So again, if the eyes are, and vision is an important part of feed detection, then eyes tend to be large and they do tend to be frontally oriented. If you're a species that detects your prey primarily through mechanoreception, the lateral line will be very well developed. So again, if you're a species that inhabits environments where the water may be very turbid, or for whatever reason you don't have the ability to see your prey or potential predators very well, the development of a lateral line that will allow you to sense them through vibrations or pressure waves in the water may be very important. And again, in the species that rely upon that, that lateral line will be much more prominent and well developed. Chemoreception is another potential strategy for detecting food as well as other things in the environment. For those species that rely upon this strategy for food detection, you will tend to see a great deal of development in those chemoreceptive structures. So the nares or the rosettes or the nostrils of the fish may be enlarged, they may be exaggerated. The fish do tend to have barbels that are going to hang down. This is particularly true in benthic feeders. It's important to recognize too that fish do have taste buds, but they're not the same as the taste buds that we have. In terrestrial animals, the processes of scent detection, smelling, and taste detection are very separate. We taste things in our mouth that are dissolved in saliva, and we smell things that are in the air, in the ambient air around us, that are volatile. In the case of fish, they are both tasting and smelling things in the same medium, so it's harder to differentiate between those two senses of smell and taste. They do have separate types of sensory cells, so there are taste buds and there are olfactory or smelling types of cells, but they aren't exclusively located in one place. So depending upon the species, they may have concentrations of these olfactory sensory cells, taste buds, on their barbels, inside their mouth certainly, but they may also have them on the surface of the body, on the opercula, for example. So chemoreception is certainly important, especially in those species where mechanoreception or certainly vision may be less valuable as a sense. Electroreception is, of course, another type of sensory system that's hard to, it's hard for humans to kind of understand because it's not one that we have. But for some fishes, they can use electrical fields essentially as a type of sonar to feel or sense the environment around them in the context of electrical impulses that are generated by other organisms. So, of course, the nervous system of any organism is essentially an electrical system, and it generates electrical charges. That's what these nervous impulses largely are. And so because of that electrical system, they can be sensed. And you can see these ampullae of Lorenzini, the little black, they almost look like little blackheads, the little pits on the nose of this shark. Those function to help amplify and sense those electrical signals. Again, this may be especially important in environments where the other senses may be less important or maybe not as important at all times. Manipulation is another form of feeding behavior. It's somewhat less common in fish. Most fish do swallow their prey whole, but manipulation, so biting, rasping, masticating, or chewing your food, is something that's done for species. They tend to be more specialized types of feeders. Certainly sharks that are feeding upon large prey items like seals that are maybe too large for them to swallow whole may bite and tear pieces of their prey away from the prey item so that they can swallow it. Mastication, chewing, you can see the pacu shown here that has what has essentially molars in the front of its mouth for chewing and crushing seeds. Rasping or scraping is something that's pretty common in fish with a parasitic aspect to their feeding behavior, like lamprey. Parrotfish, of course, are using their teeth to scrape corals from their hard structures. So again, it's something that some fish do, but it is less common in the grand scheme of all the different types of feeding behaviors that fish engage in. Okay, so we have detected our food through some sensory system, through vision, mechanoreception, chemoreception, or electroreception. We have acquired our food through ram feeding, suction feeding, some sort of manipulation, and now it is going to be entering the body. It's going to be entering the alimentary canal. And so now it's time to talk about the different components of the alimentary canal and the gastrointestinal system, and how those different elements function to help fish digest and absorb nutrients. So the alimentary canal is essentially the mouth to the anus and the tube-like structure that connects the two. So it's going to be the mouth, the esophagus, the stomach if present, the pyloric ceca if present, the intestine, and then the anus. That is a part of the gastrointestinal system, but the alimentary canal is just that. It's a part of the gastrointestinal system. The gastrointestinal system itself includes lots of other organs and tissues that are involved in digestion and metabolism, but aren't a part of that one-way street, that tube between the mouth and the anus. So, for example, the gallbladder is a significant component of the gastrointestinal system. This is going to be producing enzymes and emulsifying agents to facilitate digestion, but it's not strictly a part of that tube. The pancreatic tissue is going to be producing hormones that are involved in metabolism of nutrients. It's also going to be producing digestive enzymes, but it's not a part of that alimentary canal. So it's important to differentiate between those two elements. The alimentary canal, which is the tube between the mouth and the anus and everything in between, and then the larger gastrointestinal system, which includes the alimentary canal, as well as these other supporting organs and tissues that facilitate the digestive and metabolic processes of the fish. So we're going to go through each of these components and talk a little bit about their function, as well as the variation that's observed in that structure. So the first part of this, of course, is the mouth or the buccal cavity. Its function is, of course, for acquiring food. It may be involved in some mechanical breakdown of food. As we go through the digestive process, you'll see that it's essentially all about taking a structure that is very complex and breaking it down into its simple pieces and parts that can then be absorbed by the fish and then reassembled in a manner that suits their particular physiological needs. So one of the first steps in that breakdown of a complex structure into a more simple structure is mechanical digestion, taking a large complex structure and breaking it into smaller pieces. For those species that engage in manipulation or mastication, chewing, the mouth, of course, is going to be a part of that process. But again, for most species, they are not going to be engaging in manipulation or chewing of their food, and so the function in that case is largely just to release mucus to facilitate swallowing. There's a large variation in the different types of mouths that can be seen in fish. You can see several different examples here. There's variation in gape size, which is essentially the distance between the sides of the mouth. How big is the mouth relative to body size? That's going to limit what you can and cannot consume. Where is the shape? What is the shape and where is the location of the mouth? You can see certainly a lot of variation here in the shape of the mouth. That narrow tubular mouth is going to allow for that species to acquire food in small crevices and cracks. But the location is very important too. If the mouth tends to be at the bottom of the head, that's a good indication that the species is a bottom feeder. If the mouth has an upward orientation, they may be feeding from below and attacking prey that are swimming above them. So again, knowing a little bit about just the morphology of the mouth can tell you a lot about the types of prey items that organism or that fish is likely to be consuming, and then in turn, based on what you think they're consuming, as you'll see, that will dictate what the rest of their gastrointestinal system looks like. Of course, depending upon the species, gill rakers may be present to help retain particulate material in the mouth so that it can be swallowed, or for those species that don't consume particulate food, just to flush that material from the mouth and get it out of the gill and opercular area. Teeth, of course, are going to be important for those species that are engaged in some sort of manipulation. And keep in mind that teeth can include not just teeth that we normally think of being in the mouth, but also pharyngeal teeth that are located closer to the esophagus. So again, these teeth and gill rakers, their function is to grasp or retain that prey. Depending upon the type of tooth that's present, it may be important in terms of chewing, mechanical breakdown of that prey item, or in the case of gill rakers, if they're very finely spaced, essentially their function is going to be filtration, so to retain those small particulate food items. The esophagus is essentially the very beginning of the alimentary canal proper. Usually we think of it as being the mouth to the anus and everything in between, but the esophagus is the true beginning of the alimentary canal. It's essentially just a gateway that's going to retain material in the stomach that should stay in the stomach and prevent it from coming back out. So it's a short structure in fish. It's elastic and muscular to allow large food items to be swallowed, but then to be retained in that stomach. There's actually a typo here on this slide. Its function is not the start of chemical digestion. It is the gateway to chemical digestion. We'll talk about chemical digestion a little bit more. That occurs exclusively in the stomach, so immediately beyond the esophagus is where that chemical digestion starts. So this is the start of that process in the sense that it's the gateway to it. So once you get past the esophagus, then chemical digestion can begin. Its function may be to retain or store food, but keep in mind that this is in a very short-term sense. The food's only going to be in the esophagus when it's essentially in the process of being swallowed. So it's not like they will hold food in their esophagus for a long period of time before it actually enters the stomach. The stomach itself functions, again, largely in chemical digestion. So we're taking a complex food item and breaking it down into smaller pieces and parts. As we'll see later on, one of the best ways to take a protein and begin to unravel it and digest it is to treat it with acid. This helps to denature proteins and facilitate their digestion. So the function of the stomach is chemical breakdown of all different types of nutrients, but primarily protein. So if protein is a very important part of the diet of the fish, they will tend to have a large, well-developed stomach. Now, not all species have stomachs, as we'll talk about, but if the fish has a stomach, that can tell you that they do consume a large amount of protein in their typical diet. So the stomach, because it's involved in this chemical digestion, it has what are called oxynticopeptic cells. These are cells that release hydrochloric acid, which of course is going to be the acid that is going to denature those proteins, and also pepsinogen. Pepsinogen is an inactive form of an enzyme called pepsin. So pepsinogen is released. When pepsinogen interacts with acid, when the pH is low, it transforms into its active form, pepsin. Pepsin is a protease, meaning that it's an enzyme that breaks down proteins. So between these two things, hydrochloric acid and pepsin, it really does start to attack those proteins, those complex proteins that are present, and begin to make them a little bit simpler so that subsequent protein digestion can occur more easily. Now, another function of the stomach is essentially storage. The stomach may hold as much as 50% of the body mass of some fish, so large food items can be consumed, large meals can be consumed, and then held in the stomach until they are fully digested, and so it's going to be holding those solid food items until they can be broken down to where there are smaller particulate material, or in some cases dissolved material, that can then enter into the rest of the intestine. So that food can be held there, stored there, until it is fully digested in a chemical sense, and then can move on to the next phase. As I said, there's a lot of diversity in the morphology of fish. There is diversity in the shape of the stomach. So there are sac-shaped stomachs that are common to species like catfish, Y-shaped stomachs that are common to salmonids, J-shaped that are observed in species like milkfish, and then there's the other option of a straight or essentially an absent stomach. These are referred to as agastric fishes, without a stomach. So carp are this way, larval fish, certain types of microphagous fish, so fish that consume very small particulate materials. These are species that, again, do not consume a large amount of protein or complex proteins. Therefore, their need for chemical digestion, which primarily targets proteins, is limited. And so with very little need for chemical digestion, they don't necessarily need to have that structure or expend the energy to maintain it. So they don't have one. So here's a few examples of the gastrointestinal tracts of fish with the different components, the pharynx, the esophagus, essentially the throat of the fish labeled as number 1 and 2, the stomach labeled number 3. You can see the different morphologies that are shown here. Number 4 is referencing the absorptive surfaces. So this is essentially the intestine as well as some other structures that we'll talk about, like the spiral valve, which has been present in some species to increase surface area. You can also see number 6, pyloric ceca. These are, again, another absorptive surface that are trying to facilitate increased total absorptive capacity. So a lot of diversity based on what these species are actually consuming. There's also diversity in terms of the overall length of the gastrointestinal system. So you can see we've got surgeonfish, parrotfish, mullet, sea chubs. Generally speaking, relative to terrestrial animals, the gastrointestinal system, or rather, I misspoke, the alimentary canal of fishes tends to be fairly short. They don't have a lot of time to absorb nutrients from their food resources. But that said, there's still variation within that. So overall, fish tend to have fairly short intestinal tracts, but there is variation within that. Generally speaking, the longer the intestinal tract, the more plant-type material or less digestible material the fish tends to consume in their diet. If it's a fish that exclusively consumes other fish, for example, that's a very highly digestible protein-based food, the gastrointestinal tract will be short. If they consume a lot of plant material, like a tilapia, their gastrointestinal tract will be much longer. Before we talk too much about the intestine, I want to mention the pyloric ceca. These are finger-like projections from the anterior intestine. So not all fish have them, but for the fish that do have them, they will be present just downstream from the stomach. So you can see two examples here. The top one is a trout. The bottom one, I don't recall the species, but you can see that the trout itself has lots and lots of very fine pyloric ceca, whereas the species in the bottom here has only a couple. They tend to be a little bit larger and less complex. Generally speaking, the function of pyloric ceca is to increase surface area of the intestine. By increasing surface area, you're providing more absorptive surfaces. You're providing the fish with greater capacity to absorb nutrients from the food, or the digesta, as it's referred to at this point, before it passes out through the anus and is lost. By increasing that absorptive capacity, you can ensure that all of the nutrients are absorbed from the food, in spite of the fact that the intestinal lengths in fish do tend to be somewhat short. So increasing surface area increases absorptive capacity. So if you see more pyloric ceca, that is representative of more surface area. But it's important to recognize that pyloric ceca provides surface area, but specifically it tends to be absorptive surfaces that focus on lipid digestion and absorption. So if you see a lot of pyloric ceca, like you do in the case of the trout here in the upper figure, that tends to tell you that the types of lipids that the fish consumes tend to be more complex. So it's a species that consumes a fair amount of lipid in the natural diet, and that lipid tends to be structurally complex and a little bit more difficult to digest. In the case of trout, they're consuming primarily insects in their and certain other types of crustaceans, but largely insects in their natural diet. Insects tend to have very complex lipids like waxes present in them. these are not as easy to digest. And so having that additional pyloric ceca surface area, having the additional enzymes that are present in those structures facilitates that digestive process. So if you see a lot of pyloric ceca, it should tell you two things. It's a species that consumes a fair amount of lipid, and number two, much of that lipid is somewhat complex and more difficult to digest than simple triglycerides, for example, that would be present in most types of animal prey. Spiral valves are another structure that are similar to pyloric ceca in that they serve to increase surface area. They are present in chondrichthyans, sharks, like you see here. It's inside the intestine. You can see it's a spiral-shaped structure that goes through kind of the internal portion of the intestine. Again, it is functioning to increase surface area. It's not really known whether it has any specific function with respect to lipid digestion, for example, but it certainly does have that same surface area attribute to it. So it's going to increase total absorptive capacity and ensure that as much of the nutritional value in that digesta is absorbed before the digesta is released as feces. We've talked about the intestine a little bit already, but we're going to talk about it a little bit more. This is the site of further digestion of nutrients and nutrient uptake. So whereas chemical digestion was occurring exclusively in the stomach, now in the intestine we're not talking about chemical digestion, we're talking about enzymatic digestion. So the intestine itself is those cells are producing enzymes. Other types of enzymes are also being produced elsewhere in the body and delivered to the intestine to facilitate further processing and breakdown of complex nutrients so that they are broken down to their smallest constituent parts. They're small enough to cross the intestinal wall. So what you see in the upper figure here is a cross-section of the intestine, what's called the brush border. And you can see that there are, first of all, two large, what are called primary villi, and then lots of microvilli, small finger-like projections that are extending from that primary surface. As you might imagine, the purpose of these villi is to increase surface area. The more surface area you have in the intestine, the more capacity you have for absorbing nutrients. So the whole purpose of this structure is to facilitate digestion and nutrient uptake, and it is designed for that process. That said, there is some variation in what you see. We've already talked about how the intestinal lengths in fish tend to be shorter than what you see in terrestrial animals, but there is still variation within that. So if you see a long and thin-walled intestine, that tells you that both the uptake rate and the evacuation rate are relatively quick. So the structure, or rather the wall or the brush border of the intestine is thin, so it's easy for material to be to cross that intestinal wall and be absorbed. So that uptake rate is fairly quick. But the length of the intestine is what tells you that the evacuation rate is pretty quick. This is telling you that material, digesta, is going to be moving through the gastrointestinal system, through the alimentary canal relatively quickly, and the fish is compensating for that rapid evacuation rate by having a longer intestine. By having a longer intestine, they're essentially buying themselves a little bit more time to ensure that all of those nutrients are absorbed from the digesta before it's released as feces. So if you see a long and thin-walled intestine, it's a good indicator that this is a species that's consuming a diet that is not especially rich in protein or other nutrients. So carp or tilapia would be a good example here. If you see an intestine that is short and has many ceca, that's telling you that there isn't a whole lot of retention time here, and so the fish is trying to compensate for that short retention time and that short intestine by having many of those pyloric ceca to increase surface area and ensure that any of the nutrients that are available can be absorbed prior to evacuation. As we already discussed with respect to those pyloric ceca, if there's lots of them, that's a good indicator as well that the diet of this fish tends to have a fair amount of lipid and complex lipid especially in it. Now on the other hand, if you see a short intestine but it's relatively thick-walled and doesn't have many or any pyloric ceca, that's a good indication that the uptake rate may be fairly slow and the evacuation rate may be slow. A good example here would be something like a largemouth bass. Largemouth bass are consuming other fishes exclusively. And so that large prey item is going to sit in the stomach and be chemically digested until it's essentially liquefied. An intact prey fish can't pass through into the intestine in a solid form. It has to be liquefied, essentially, mechanically and chemically broken down prior to entry into the intestine. So that means that that prey item is going to sit in the stomach for a fair amount of time. The amount of time it's going to take for that prey item to pass fully through the alimentary canal is going to be relatively lengthy. So that evacuation time is fairly slow if you think about the total amount of time that the prey item spends in the alimentary canal. Not just the intestine, but the stomach as well. So it's going to take a fair amount of time. But again, they don't need to have a lot of surface area. They can get away with having a short intestine because by the time any material is released from the stomach, it has been thoroughly digested, both mechanically and chemically, largely chemically, by that acid that's in the stomach. So once it enters the intestine, it's liquefied, it's well broken down, it's easy to absorb, so they don't necessarily have the need for a large number of pyloric ceca or spiral valve, etc., to increase that surface area. So again, what you see in terms of the intestinal morphology will tell you a lot about what that species is consuming and what its feeding behavior might be in general. Finally, we reach the rectum, which is the most posterior portion of the intestine. There isn't a huge amount of functionality here. The rectum is essentially holding the digestive prior to evacuation or defecation. There may be some final reabsorption of water and certain nutrients occurring here, but this is essentially where the excretory processes are going to be dominating. So in those species that have a rectal gland, for example, this is where they're going to be introducing those excess ions into the digesta, ultimately, which will be becoming feces prior to excretion. I want to talk a little bit about this functional blueprint for the gastrointestinal system or the alimentary canal. We're going to talk about it in terms of essentially what the fish needs and that determining what the fish has in terms of morphology and function. So this is essentially a discussion of beau plan, like we've had earlier in the class, form and function together. So if mechanical breakdown is going to be an important part of the total digestive process of fish, that's going to indicate a certain type of food or feeding behavior. So that will be driven by the size of the food item. So if the food item is very large, too large for the organism to be consumed intact by the fish, or if it's very structurally complex. So for example, it's a crustacean and it has this hard exoskeleton which must be broken down to liberate the nutrients within. That's going to drive the need for teeth or gizzards to help grind and break down and mechanically break down that food item. So again, the type of food and the feeding behavior is going to drive what the fish needs to have in terms of teeth, gizzards, what have you. Chemical breakdown, the importance of chemical digestion, is also going to be driven by the food itself. So first of all, how available are nutrients in the food? And what is the composition of that food? If it's primarily plant material without a lot of protein content, there really isn't going to be a whole lot of need for chemical breakdown because carbohydrates are not terribly vulnerable to acidic digestion. If, on the other hand, the composition of the food is very high in protein, so you're consuming other animals, other fish, protein is very vulnerable to acidic digestion. That would indicate a need for chemical breakdown to improve those substrates, those proteins, for further digestion by enzymes. And so that's going to drive whether you have any kind of, whether you have a true gastric stomach or whether you don't have a stomach at all. Keep in mind, again, that chemical digestion only occurs in the stomach. So if the fish has a stomach, they engage in chemical digestion. If they don't engage in chemical digestion. Now this is actually a series of slides that I took from another lecture on endocrinology and talking about gastrointestinal tracts in other species, including ruminants. So this mentions the question here about pre-gastric fermentation. This is not something that occurs in fish because they are monogastrics, but it's something that is a part of the overall strategy for ruminants. So for the purposes of fish physiology, you don't have to worry about pre-gastric fermentation. That's something that occurs in ruminants exclusively. So going back to this question of chemical breakdown, again, the composition of the food is going to drive the need or lack thereof for acidic digestion, which will then drive the need for the presence of a true gastric stomach or no stomach at all. Enzymatic digestion is again going to be driven by nutrient composition and complexity. So if you consume a lot of protein, a lot of different types of proteins, you're going to need a lot of different types of proteases, enzymes that break down proteins. If you consume a lot of lipids, especially complex lipids, you're going to need to have a lot of enzymes that break down those types of molecules. So you'll need a lot of lipases. Carbohydrates are going to be enzymes that break down carbohydrates. It's also important to recognize that the need for these enzymes is going to be largely supplied by the fish itself. So the fish is going to produce its own proteases, lipases, etc. But the microbiome, or the microbes, the microbial community that colonizes the intestine of fishes, can also be a source of some enzymes. So microbially derived enzymes may be especially important for those species like tilapia or carp that are consuming a diet that would otherwise not be terribly well digested by the fish based on the enzymes that it itself produces. So again, what the type of food is, the nutrient composition, and its complexity is going to drive the types of enzyme systems that the fish either produces or is otherwise able to acquire, for example, through the microbiome. The absorptive processes are going to be a function of nutrient density, the evacuation rate, and the surface area needs. All of those different questions are going to influence the intestinal length and the thickness of the wall. Hindgut fermentation. Now this is fermentation after chemical digestion. This is essentially digestion by the microbiome. This is something that occurs in fish. We talked about how pre-gastric fermentation doesn't occur in fish. That's something that happens in ruminants. But this type of post-chemical digestion fermentation, or hindgut fermentation, or digestion by the microbes that live in the intestine, this can be very important in certain types of species. Emulsification. This is the process of taking lipid, which would otherwise not be soluble in the aqueous environment of the intestine, and ensuring that it does become emulsified and then can be absorbed. The need for emulsifiers is, of course, going to be driven by the presence of a lot of lipid in the diet. Passive absorption versus transport or facilitated uptake. This is going to, again, depend upon the types of nutrients that fish is consuming. So once again, form and function are going together. If you're consuming a very nutrient-dense food that requires a fair amount of emulsification because it has a lot of lipid in it, that's going to drive the need for lots of lipases, emulsifiers, and perhaps in some cases transport or facilitated uptake to ensure that all those nutrients are actively absorbed, and you're not relying on passive processes because those simply might not be fast enough to acquire all the nutrients as quickly as you need to. Distribution and metabolism of resources is, of course, going to be driven by the state of the fish. So are we talking about a larval fish, an on-growing fish, a fish that's entering reproductive maturity? Its developmental state is going to determine how it's going to be metabolizing those nutrient resources that are acquired. Its bioenergetic state will also be a large driving force in that. Is this a fish that has been consuming food on a regular basis? In which case, it's probably in an anabolic state, a growth state. Are we talking about instead a fish that has not been consuming food regularly and is in a period of starvation? It's likely going to be in a catabolic state, where it's breaking down its bodily resources to fuel energy demands. So bioenergetic state, developmental state are going to be related to whether or not the fish is undergoing catabolism or anabolism. Tissue demand for different types of resources is going to influence how nutrients are trafficked throughout the body. Oxygen flux and intermediary metabolism are two things that we'll talk about a little bit more later on in the course. This is again going to be a factor in terms of how nutrients that are acquired ultimately are distributed to the different tissues and how they are utilized for either growth processes, anabolic processes, or catabolic processes for energy production. So again, mechanical breakdown, chemical breakdown, enzymatic breakdown, all these digestive processes are going to facilitate, hopefully, adequate nutrient absorption, and then those nutrients, once absorbed, are going to be distributed and utilized by the body depending upon the state that it's currently in. So let's talk a little bit more about digestion and absorption. We're going to talk about these processes in more detail. You've already heard where it happens, but now we're going to talk about how it happens. So we're going to talk about digestion in general, then we're going to go into both chemical and enzymatic digestion of proteins, lipids, and carbs, and then we'll talk briefly about active and passive absorptive processes. So again, chemical digestion is occurring in the stomach. It says primarily here it's almost exclusively. Chemical digestion is the process of treating food items, whether they contain protein or what have you, with acid. Now, acid is going to denature protein, it's going to unravel protein. It may help to emulsify lipid to a certain extent. It doesn't have a lot of activity with respect to carbohydrates. So if you're talking about a species that consumes a fair amount of protein and a certain amount of lipid, chemical digestion is going to be pretty important. Of course, this low pH, this acidic pH in the stomach is going to increase the activity of acidic enzymes. There are enzymes that are present only in the stomach, and they cannot function at a pH other than what they find in the stomach. So once they pass into the intestine and are neutralized and become more alkaline, those enzymes no longer function. So chemical digestion is treatment with acid. It is primarily functioning to denature protein, unravel those proteins, and make them more accessible to enzymes for further digestion. And it, again, is occurring exclusively in the stomach. Chemical digestion, it's important to note, does ultimately stimulate the release of something called secretin. Secretin is a hormone that is going to be involved in neutralization of that acidic digesta once it reaches the intestine. So the chemical digestive process, of course, is going to initiate the further digestive processes that will occur in the stomach. All these processes are going to be linked. It's important, I think, at this point to talk briefly about enzymes. I think everyone is probably familiar with what an enzyme does, but enzymes are, just to make sure that we're all on the same page, enzymes are chemical catalysts that are not consumed during a reaction. So they are going to be facilitating a chemical process, making it faster, easier, etc., but they are not, as it says here, consumed or used up during that process. They can then facilitate the same reaction over and over again. Now, oftentimes, the enzymes are named for their function and then they're given the suffix ase. So when we talk about a lipase, this is an enzyme that breaks down lipid. When we talk about proteases, those are enzymes that break down proteins. So that's generally the case, but not always. There are, of course, exceptions to the rule. Now, if you have a suffix that's ogen, that means it's an enzyme, but it's in its inactivated form. So when we talk about pepsinogen, that is the inactive form of pepsin. Pepsin is a protease. So it doesn't have that ase suffix. This is one of our exceptions to that rule. But in its pepsinogen form, it's not active. But in the case of this particular enzyme, when it is acidified in the stomach through chemical digestive processes, it becomes activated, it turns into pepsin, and then begins to have its protease function. So when you see these different types of suffixes and different enzymes, enzyme names, hopefully this will give you some ability to identify, yes, that is an enzyme, it ends with the suffix ase, or, well, it's ending with the suffix ogen, but that's still an enzyme, it's just not in its active form. So let's talk about this through this process one more time here about what's going to be happening at each stage. Now, of course, this is not the gastrointestinal system of fish. This is a diagram from humans, but the function is largely the same. So we begin with the mouth. We have salivary glands present. This is going to be functioning in the preliminary digestive processes and then lubrication. So we talked about how for fish that engage in manipulation, they may be mechanically breaking down their food a bit, initiating the digestive process through mechanical breakdown, but mostly what's happening in the mouth and the esophagus is the production of mucus to lubricate that food item and facilitate swallowing. Once we get into the stomach, this is where chemical digestion is going to be occurring. So we have acidic lysis of cells that exist in the food item. And again, this is mostly going to be addressing proteins. It may have some benefit in terms of digesting lipids, not a lot of benefit in terms of carbohydrates. So mostly protein is what's happening and being addressed in the stomach itself. Then we enter into the intestine. This is where you have lipid digestion, carbohydrate digestion, and further protein digestion, but this is in the context of enzymes. So chemical digestion is stopped when the digestion moves from the stomach into the intestine. We switch from chemical digestion into enzymatic digestion. At the same time that we have these digestive processes going in the intestine, we also have nutrient absorption occurring. So as those nutrients are broken down into small enough pieces and parts, they are absorbed across the intestinal border, across that wall. To facilitate these digestive processes and absorption, you have bile and various types of enzymes, including lipases, being produced by the liver and the gallbladder. And so those are going to be released and introduced into the intestine to, again, facilitate emulsification of lipids and to support lipid digestion. At the same time, the pancreatic tissues are going to be producing a variety of different types of digestive enzymes, lipases, proteases, carbohydrases. It's going to be releasing those into the intestine as well, again, to facilitate this digestive process. One thing I want to note here on this figure, this is a diagram, again, from a human, so it's indicating a discrete pancreas. Fish don't have a discrete organ that you can identify when you dissect them. Here's the pancreas. What they have is diffuse pancreatic tissue that kind of surrounds the intestine. So they have pancreatic tissue that has the same function as a true pancreas, but it doesn't exist as a discrete organ in fish. Okay, so the pancreas is going to be releasing and producing digestive enzymes. The liver is going to be producing enzymes and introducing those as well. The gallbladder is going to be releasing bile. This is going to help with that digestive process. The other thing that's going to be occurring at the same time is you have the release of bicarbonate ions into the intestine. These bicarbonate ions are produced by the pancreatic tissue and they're released into the intestine to neutralize that acidic material. So when it's in the stomach, the pH may be as low as 4. This is, again, to facilitate those acid-active enzymes, and again, the acid itself is going to facilitate protein digestion. That's happening in the stomach, that's a good thing. But once that acidic material moves into the intestine, it has to be neutralized because the enzymes that are present there, as well as the delicate structures that are present there, will be destroyed by acid. So the pancreas is going to be producing that bicarbonate that's going to neutralize that acidic material, bring it into a neutral or slightly alkaline state, and then these other enzymes and processes can occur. Then we get into the lower portions of the intestine, into the rectum. Again, this is where most of the microbes that represent the microbiome of fish are going to be living in that intestinal area, particularly the posterior intestine. So you may have some microbial fermentation occurring that's going to further process any of that otherwise indigestible material. Again, you're going to have some nutrient absorption, but then as you release, or rather as you reach that rectum, it will be largely any kind of moisture absorption, water reabsorption, lubrication, release of mucus, ultimately to facilitate defecation. So this is, again, just a review of the different processes that are occurring and where they're occurring in the body of fish. So let's step through this kind of generalized digestive process here. So this is a view from below. You can see the stomach, the pancreas, and the gallbladder of fish that are highlighted here. So the first thing that happens in terms of the digestive process is the consumption of a food item is going to distend the stomach. So the fish eats something, its stomach swells, and the swelling of that stomach is going to trigger the release of a hormone called gastrin. Gastrin is a hormone that's produced by the stomach itself, and gastrin is going to function exclusively to trigger those oxynticopeptic cells that we discussed to get them to release hydrochloric acid and pepsinogen. Okay, so it's the distension of the stomach. The stomach gets bigger when there's a food item in it. That sends nervous stimuli to the stomach itself saying, hey, we've got a food item here. We need to start the digestive process. In response, the stomach produces gastrin, and gastrin, as a hormone, will trigger the oxynticopeptic cells to release hydrochloric acid and the acid functional enzymes. So they're releasing that pepsin, they're releasing the hydrochloric acid, the digestive becomes acidified, and the process of chemical digestion begins. Once that acidified material has been fully digested and can be released from the stomach, now you have low pH, very acidic digestive moving into the anterior intestine. The low pH of that material in the anterior intestine will trigger the release of secretin. So secretin is going to be produced. It's another hormone that is going to be triggering the release of bicarbonate ions and enzymes from the pancreas. So the acidified digesta hits the intestine. The intestine says, whoa, this is acidified material. We can't function with this. We have to neutralize this acidified material. So secretin is released, acts upon the pancreas, and tells the pancreas to start releasing bicarbonate ions to neutralize the digesta and also to initiate enzyme production. Okay, so this hormone is produced, this secretin is produced by the anterior intestine, acts upon the pancreas to get the pancreas to neutralize that material in the intestine. Now, as this process continues, as the digestive process continues in the intestine, we've neutralized the digesta, so now the various enzymes that are being produced and delivered to the intestine, as well as the enzymes that are produced by the intestine itself, they are going to begin to break down lipids. As the presence of free fatty acids and free amino acids increases in the intestine, that is the next trigger. So as the nutrients become further broken down by enzymes, they're going to release these constituents of those nutrients. So lipids will be broken down into free fatty acids. Proteins will be broken down into free amino acids. As the concentrations of those free amino acids and free fatty acids increases, that's the trigger for cholecystokinin to be produced. Cholecystokinin is the third hormone in this generalized digestive process. It acts upon the gallbladder, causing it to contract. That contraction of the gallbladder is going to release and produce, or it's not really releasing anything, it's just the contraction of the gallbladder is going to facilitate bile being delivered to the intestine. That bile is going to act as an emulsifier to help facilitate that lipid and ensure that all of those free fatty acids that are being produced are dissolved in the aqueous environment of the intestine and then can be effectively absorbed. So this is the generalized process, and I want everyone to know these three hormones. Gastrin, which is produced by the stomach, acts upon the stomach to trigger the release of hydrochloric acid and pepsin. That's going to initiate that chemical digestive process. Then the movement of acidified digesta into the anterior intestine is going to trigger the release of secretin by the anterior intestine to act upon the pancreatic tissue to get the pancreas to release bicarb for neutralization and to release enzymes to further spur the digestive process. Then, as those digestive processes continue, the increasing abundance of free fatty acids and free amino acids as a result of those digestive processes, the presence of those nutrients is going to trigger the intestinal tissues to produce cholecystokinin, which will then act upon the gallbladder, causing the gallbladder to contract and for bile to be released into the intestine. Again, that's going to facilitate those lipid-soluble nutrients, allowing them to be fully absorbed. So I hope that everyone understands this general process. I guarantee there will be a question about this process on the exam. Let's talk a little bit about digestion of carbohydrates. So when we talk about carbohydrates, we're talking about a variety of different molecules that are essentially linkages of glucose and other simple sugars. So for these nutrients to be digested, these complex carbohydrates have to be broken down into their simple sugar constituents. Now, not all carbohydrates can be digested by fish. So if we're talking about a simple starch, which is shown here at the top, those have linkages between the glucose molecules that can be effectively digested. But when we talk about cellulose, for example, it's also a simple chain of these linkages between glucose molecules, but those particular types of linkages are a slightly different type of bond that cannot be broken by anything other than a cellulase. Fish don't produce cellulases. And so this is what's going to determine what is a digestible carbohydrate and isn't a digestible carbohydrate. Does the fish have or produce an enzyme that's appropriate to cleave these bonds between the simple sugars? They cannot be absorbed unless they are in a very simple form. So a complex, a full starch molecule, even a simple starch, is going to be too large to absorb in the intestine. It has to be broken down to those individual glucose molecules. Same thing with all of these polysaccharides, but they have to be, the bonds between those simple sugars have to be digestible. They have to be able to be broken by enzymes that either the fish itself produces or organisms that live in the gut of the fish produce. So generally speaking, digestion of carbohydrates would begin in the mouth with any salivary carbohydrates. Now, it may be that fish produce some of these carbohydrates, but because of all the water that's moving through their mouth, it's going to be diluted substantially, and so there isn't going to be a large amount of digestion occurring in the mouth or the esophagus with respect to carbohydrates or anything else for that matter. This is really mechanical digestion that occurs here. So if there's any kind of mechanical digestion, that's going to help with carbohydrates and everything else. Otherwise, there's going to be very little digestion occurring until you get to the small intestine. So the stomach acids tend to stop any kind of carbohydrate digestion that might be occurring. Carbohydrases tend to be active at neutral or alkaline pHs. So in the stomach, the pH is very acidic. Those enzymes can't function. But once you get into the small intestine, various types of carbohydrates, including amylases that are produced by the pancreatic tissues, as well as carbohydrates that are produced by the intestine itself, these will continue to break down polysaccharides to the extent that they can. Now, there are going to be some enzymes that are actually associated with the gut mucosa. So I think generally speaking, we think of these enzymes as functioning in a dissolved liquid form. And for the most part, they do. They're functioning in what's called the lumen of the intestine, or the space within the intestine. So those enzymes are solubilized and they're dissolved within that liquid material, and that's how they're functioning. But there are other enzymes that are anchored to the gut mucosa, or they're anchored to that internal lining of the intestine. And so these can further process the more simple polysaccharides, breaking them down into those simple sugars, including glucose, and then ultimately helping to absorb them. So there's both luminal digestion of carbohydrates, something that's occurring that's kind of loose and dissolved in the liquid material in the gut, and then you have mucosal associated enzymatic digestion by these enzymes that are anchored in that intestinal lining. With respect to proteins, keep in mind that proteins are, of course, comprised of amino acids and are arranged into these four levels of structural arrangement. So we have the primary structure at the top. This is essentially the chain of amino acids that are bound together. You have the secondary structure. So we have beta-pleated sheets or alpha helices, this kind of primary shape to the chains of amino acids to the polypeptides. These beta-pleated sheets and alpha helices are then arranged in different configurations to form the tertiary structure of the complete protein, and then you can have a quaternary structure as well, where you have multiple proteins bound together. A good example of a protein with a quaternary structure would be hemoglobin, which of course is comprised of those four different groups that are bound together, the beta groups and the alpha groups. So we have to take proteins from this quaternary structure, if present, all the way down to that primary structure and then break them into individual amino acids in order to be absorbed. So we have to take this very complex structure and take it all the way down to very simple peptides, either 2 or 3 amino acids linked together and that's it, or preferably all the way down to individual amino acids for absorption. So this protein digestion is going to be initiated in the stomach by that chemical digestive process. So the hydrochloric acid that's produced by the oxynticopeptic cells, that's going to denature proteins. It's going to begin to unfold that quaternary and tertiary structure. That's going to make those proteins then much more accessible to the initial proteases that will attack them. So pepsinogens are released by the oxynticopeptic cells as well, and those pepsinogens become activated by the low pH. So they start with their full function. They become functional pepsins. And then in that sense, they assume what's called an endopeptidase function. So there are 2 major types of proteases, endopeptidases and exopeptidases. These descriptions refer to where the enzymes cleave amino acids. So if you're talking about an endopeptidase, it will go and cut a chain of amino acids in the middle. So that's what the endo refers to, inside. So it's going to cut and cleave long chains of amino acids by cutting them somewhere in the middle, making smaller amino acid chains. Exopeptidases, on the other hand, they work from the outside in. So they will attack the one end or another end of a peptide chain and remove 1 or 2 or maybe 3 amino acids at a time. In fish and in other organisms, you have both endopeptidases and exopeptidases. Pepsins are the first of the endopeptidases. So these take these very long chains of amino acids that have been unrolled and unfurled by the denaturing process, by the hydrochloric acid, and they go into the middle and start snipping them. So we start with long chains of amino acids, very long peptides, and then we snip those into shorter chains. Further on, once we get into the small intestine, then you have additional enzymes that are functional. So in the small intestine or the intestinal structure entirely, you have neutralization of the acidic digesta. So those pepsins will go back to a pepsinogen form because they're only active at low pH. But then you will have activation of the neutral or alkaline peptidases. These tend to be exopeptidases. So we have pancreatic trypsin, chymotrypsin, and other types of amino acids that will now work on those smaller, shorter chains of peptides that were produced by the endopeptidases, and they'll start removing individual amino acids from either end of those shorter chains. So we start with the endopeptidases, snipping the long chains into shorter chains, and then we move into the exopeptidases, which then will start removing individual amino acids from either end of those shorter chains. So all these peptidases, all these proteases are working together, but they would not be able to attack that secondary and primary structure of proteins if the proteins hadn't been denatured by the acid present in the stomach. With respect to lipids, remember that lipids include phospholipids, sterols like cholesterol, and largely triglycerides or triacylglycerides or TAGs. Just like proteins have to be broken down from their full complex structure into small dipeptides, tripeptides, or individual amino acids, lipids have to be broken down to their free fatty acid forms, glycerol molecules, etc. Now, occasionally you can have a diglyceride or a monoglyceride be small enough to be absorbed without further digestion, without further processing. But for the most part, we can think of this as being a process that we have to take these phospholipids and triglycerides, for example, remove each of those fatty acids from the chain so we have the component parts that are small enough to be absorbed. And again, this is a very similar process going from a very structurally complex lipid to individual triglycerides, phospholipids, etc., and then breaking those individual molecules down into even smaller parts for absorption. Digestion of lipids really begins in the intestine or in the pyloric ceca, really. Again, there may be some help with respect to emulsification of lipid that occurs during chemical digestion in the stomach, but lipid digestion truly doesn't really get started until you hit that small intestine. So in this case, we have a variety of enzymes that are being produced by the pancreatic tissues. So they're going to be producing a lot of lipases that break down triglycerides as well as phospholipids. That are going, again, they're going to be broken down into monoglycerides. So that's a glycerol molecule with only one fatty acid instead of three. You'll have free fatty acids, some free glycerol, etc. Keep in mind, too, that those bile salts, or the bile that's being produced and released by the gallbladder, are going to ensure that all of those different products that are produced by these enzymatic digestions are fully emulsified, dissolved in the aqueous environment of the intestine, and then ultimately absorbed. Now, these cleavage products, or the monoglycerides, free fatty acids, etc., once they're absorbed by the cells of the intestine, they may be further digested intracellularly. But that's dependent upon the product itself and the needs of the cell. So the primary process is going to be enzymatic in nature, breaking down the complex lipids into simple free fatty acids and other lipid components, and then emulsification by the bile salts to ensure effective absorption in the intestine.